This is the last of 4 separate experiments dealing with food microbiology. Previously, there was one done with standard plate count procedure where we were looking at food items such as potato salad. In this one, we're looking at milk. What we're wanting to do is see what is the bacterial load, or basically how many bacteria per milliliter are in the milk sample. Various representative samples in the processing or the production of the milk need to be tested to make sure that it is within safety limits for food quality, food safety items. So this is done, it's required by FDA. It's going to be done, like I say, in the production of the milk. You want to determine if there's a level of contamination where it's unsafe for human consumption. So how do you test it? You're going to do a serial dilution first. We know that milk is going to have some bacteria in it because it is not autoclaved. It is only pasteurized. So you're going to do a serial dilution, then you're going to follow that by doing your standard plate count. You either do a pour plate or a spread plate, and where you put the sample from the diluted samples on a plate. Remember, you will only count between 30 and 300 colonies. In whatever count number you get, then you're going to multiply that by the dilution factor to be able to relate it back to the original sample to say it has X number of bacteria per milliliter. So as I just mentioned a bit ago, milk is going to have some bacteria in it. We do not sterilize milk because the sterilization process, the heat under pressure, is going to break down the proteins in the milk. It's going to alter the taste of the milk. People want milk to taste like milk, and it's going to taste weird if it's autoclaved. So we don't sterilize, and instead we use pasteurization. There's different levels of pasteurization. What would be considered the traditional form. Then there's also ultra pasteurization. You go to a higher temperature for a certain amount, less amount of time than you would for the regular. But either way, pasteurization is going to kill some of the pathogens because you are increasing the temperature for a certain designated amount of time. But it's not going to kill everything. The idea is it kills the pathogenic microorganisms, but the non-pathogenic ones will still remain. So that's what you're trying to see, is what is that load? What is that number in the milk? Even if they're non-pathogenic, if the number is really high, you don't want to use it for human consumption. So what we do in this lab is we take a milk sample and we do our serial dilution, and then we plate it out. Usually we use a spread plate when we're doing this. And this is showing a sample where the milk was not diluted at all, and this is just solid growth on the plate. Now over here, as you can see, the dilution factor is 1 to 100,000, and by this point there's just a couple little colonies over here that you can detect. But you can see how that when you dilute it out, and so you're going to reduce the number. That's what dilution is doing. So you're going to have several different plates in a range to detect. It falls in that 30 to 300. So in this experiment, we have the undiluted, we have a 1 to 10, 1 to 100, 1 to 1,000, 1 to 10,000, and then 1 to 100,000. But ultimately, the bottom line is you're talking about food safety here. How do we guarantee that the food supply is safe for human consumption?